When people think about nucleic acids, they typically think of DNA, but there's another molecule, RNA, which is just as important. It's the one that takes the information that's being stored in DNA and sends it out to the cell so that the cell can actually use that information. DNA is a really long molecule, RNA is typically a shorter molecule, but it's just as important as DNA. It's the workers that help carry out some of the information and instructions of the DNA. And it's built together much like DNA is. So let's take a closer look. The basic building blocks that make up RNA are nucleotides, just like with DNA. Just like DNA, it has a phosphate group that gives it a strong negative charge. It has a 5-carbon sugar, sometimes called a pentose, and some kind of nitrogen-containing base or nitrogenous base. Now, one of the differences to bear in mind between DNA and RNA is what is that pentose sugar? Well, deoxyribose and ribose are the two sugars. Deoxyribose, can you guess which one uses that? You're right, DNA, which stands for deoxyribonucleic acid, while RNA, ribonucleic acid, uses ribose. And if you look at the names, they look very similar. In fact, if I cover up the deoxy, I see the word ribose. What does that mean? Well, this is ribose. Notice down here on the second carbon, there's an OH group or hydroxyl group for those who are into chemistry. If I pull off that oxygen, i.e. deduct it, then I've deoxygenated this ribose here. And look, all that's left is the hydrogen. So that's the difference between ribose and deoxyribose sugar. The other difference that you'll see in the structure of the nucleotides is that it uses the same guanine and adenine and cytosine that DNA uses, but instead of using thymine, it uses a particular kind of pyrimidine called uracil. Now, to join RNA molecules together, it works pretty much the same way as joining DNA molecules together. You take a phosphate and sugar and nitrogenous base, i.e. a nucleotide, and you bring the phosphate group of the next one in and it joins a phosphate to that sugar. And then you extend that. And so you wind up with a long strand of RNA nucleotides with their bases sticking out, with the phosphates and sugars forming the backbone of the strand. Now, you're familiar with this with DNA, and you know that DNA often twists up to form the very famous double helix. Well, RNA can do that, but because of that additional oxygen that's on this carbon right there, it tends to make it unstable for long stretches to be in a double helical form. For short portions, however, you can. And the way you can form an RNA to RNA strand or RNA to DNA strand follows the same base pairing rules that DNA does, with a little twist. Remember that RNA does not use thymine, it uses uracil. DNA, if we're binding DNA to RNA and we have an RNA adenine here, this would have to be a thymine for DNA. But if I was making an RNA to RNA where I have an adenine, I'd have to use uracil, which you'd abbreviate U. So if I have my RNA strand here, that's A, C, A, I follow the standard base pairing rules of A to T, or U, G to C. So here's the C, there's a guanine, or G. Here's an A, I put a thymine. So that's it. Pretty straightforward. It's much like DNA, just with those little differences. One mnemonic or trick to help you remember the key difference of using uracil instead of thymine is, remember, what's the abbreviation for uracil? It would be the letter U. So just think in your head, you are correct. And if you are correct.